Students, are you able to see my screen?
No, ma'am.
No, ma'am.
Okay, okay. Just a second. Now, can you see?
Yes,
Yes,
ma'am.
ma'am.
Yeah. So we all know that glycine is the one of the simplest amino acid, which is a semi-essential amino acid. So hence it has to be taken up with nutritional support, and it is one of the major inhibitory neurotransmitters. Okay, so one of the major after GABA, you can see this glycine as one of the major inhibitory neurotransmitters, which is present in your vertebrate CNS. So these synapses are called as your glycinergic synapses. Particularly, they are present in high amounts in spinal cord and brainstem, but some amount is also present in hippocampus. So glycine receptors are like they are classified under your nicotinicoid receptor superfamily, and hence it is also known as your inhibitory chloride channel. So synthesis, this is how you have the synthesis of glycine again. So structure, when we talk about receptors, it is a pentameric protein. So made up of homo-oligomers of alpha isomers. So there would be alpha 1, 2, 3, or 4. And you have beta subunits, beta 1, which is essentially the target for the receptor. Okay, so where your binding can be done. So as we have seen in case of your GABA, similarly here also your receptor is made up of 5 subunits. Okay, so you can see over here similar structure. Okay, so it is made up of 5 subunits. So 3 is alpha and 2 is beta. Okay, 2 beta subunits and 3 alpha subunits. And central portion, you can see it will form a pore. Okay, so they are presently in 4 isomers. There are different isomers which are called as your alpha 1 to 4, depending upon your which alpha isomer is binding to it. So hence it can be present as your GRA1, 2, 3, 4, and there is a single beta subunit that is B. So adult form of your these receptor is heterodimeric, that is your alpha 1 beta receptor, which is believed to have your stoichiometric proportion. That means this thing that you are talking about, this is the most common one that you have. So it is mostly composed of protein together, that is your Geoffrin protein. So this Geoffrin protein is bound to your beta subunit, which is helpful in anchoring the protein into the postsynaptic zone. Okay, so this protein, this is a transmembrane protein, that is Geoffrin, which helps us in holding these together. So these are located, these genes which are there, they are located in your chromosome 5. So we don't have to go in details of that. Let us understand the mechanism. So here also, as it is an inhibitory neurotransmitter, the mechanism is going to be same. That means it acts as ligand-gated ion channel. Okay, but sometimes very important, the glycine will also can act as an excitatory neurotransmitters during the embryonic development and around the birth. But in adults, definitely glycine is always inhibitory in action. But in embryos and during the birth, so this transition takes place. So this is due to positive chloride equilibrium potential of the postsynaptic neurons, which results in your chloride efflux upon glycine R activation, causing the depolarization. So that is going to happen in case of your embryonic development, because in the embryonic in the embryos the chloride ions which are there, it is ulta. That means you will have more amount of chloride ions inside and less amount outside. So hence, when the chloride channels open up, then there is an efflux. That means instead of coming inside, because the concentration is more inside, it will move outside. Okay, so in general, you will have the shift, the chloride equilibrium potential shifts to the negative value owing to the active chloride exclusion. So that is, you have this potassium chloride plus co-transporters which are present. So glycerin receptor activation becomes hyperpolarizing and therefore inhibitory. So let us understand what happens during this. So this is your inhibitory mechanism that happens in general. So you can see over here in that what happens is that because of your, these are your sodium potassium ATPases, and because of that, you know that the concentration of your potassium ion is always kept more inside, and sodium ions is more outside. So then you will have this different types of your co-transporters which are there. So that is called as your K plus Cl minus co-transporters, KCC2, KCC3. So what happens is with your these co-transporters, always the concentration of your like when your potassium ion is moving outside, your chloride ions are also moving outside, and that is how your chloride ions are kept like they are kept more outside the cell. Whereas if you see in this immature cells, that is in embryonic development, you see that your chloride ions are coming inside. यहाँ पे ये arrows को देखो. Here the chloride ions are going outside. Here the chloride ions are coming inside. Yes. So what happens is the concentration of your chloride ions is more in outside the cell in mature cells, whereas in immature cells the chloride ions are more inside the cell. So when your these chloride ion channels open up because of your action of your glycine, then what will happen is chloride ions move inside in mature cells, causing your hyperpolarization, whereas in your immature cells they move outside, causing your depolarization. Yes, everyone, is this very much clear to you? Understood what is happening? Yes. So it depends upon what is the concentration gradient of these ions, and depending upon that you can find out what exactly is happening when your ion channels are opening up. Okay, so the same thing I have just explained it to you. So the function of these receptors is to prevent tissue injury, to enhance antioxidant capacity, to promote the protein synthesis, wound healing. You have biosynthesis by different ways of heme, creatinine, glutathione, and regulation takes place with the help of different PTKs, that is your protein tyrosine kinases, which are there. So these are your agonists. You have taurine, which is one of the partial agonists which you have. You have beta alanine, which is again acting as your agonist. Beta alanine, L-alanine, hypotaurin, L-serine, these are all agonists. So these agonists, if you see the structure is very much similar to glycine, and that is how glycine will be helping in acting as your agonist. So in the central nervous system, there exists this strychnine sensitive glycine binding sites as well as strychnine insensitive glycine binding sites. So strychnine is nothing but it is a antagonist. Strychnine is a kind of a poison. It is an antagonist for your glycine. So these are located on your NMDA receptor complex. So NMDA receptors you will be learning when we are talking about your catecholamines. So I'm not going in detail of that. So whenever you have this strychnine sensitive glycine receptor, so which comprises of your these chloride channels, okay, and you have this strychnine insensitive, which is nothing but your mostly that is related to your different types of your metabotropic receptors which are there. Okay, so they are mostly involved in the chloride conductance, as we have already seen, and subsequent enhancement of inhibitory neurotransmitters. So the basic thing is about how your inhibition can be taken up. So these neurotransmitters would be involved. There are many studies which have shown that these neurotransmitters could be involved in schizophrenia also. So hence it is used in the treating of schizophrenia, stroke. You have benign prostatic hyperplasia and some other metabolic disorders which are there. So that's the major use that you can have of glycine in with respect to your this neurotransmitters. So as I was talking about, these are your antagonists. You have strychnine, you have brucin, you have cutin, you have bicaculin, which is caffeine, picrotoxin, pitrazepsin. So these are the different your different types of your antagonists which are present. So talking about disorders, so you have hyperplexia, which is your hereditary neuromotor disorder, where your these receptors are impaired. That means they are not working properly because of your genetic disorder. So it is a startle reaction. That means always these kids or whenever there is a small stimulus, what happens is they get startled, they get very excited. Okay, so मतलब बहुत ज़्यादा चौक जाना. Startling means what? चौंकना. So even if there is slight amount of your sound or anything, they get startled. So that's about your startle disorder. But because these receptors are not working, these inhibition does not take place. Okay, so you have a drug clonazepam, which can be used for this treatment. Then you have bovine myoclonus, which occurs, which is the same startle syndrome in myoclonus, or so that is your bovines. Okay, same thing. Again, having a problem in the alpha 1 subunit gene, which is there, and that will give rise to this particular type of your disease. Then you have non-ketotic hyperglycemia, which is an autosomal disorder. So where the enzyme which is responsible for this breaking down of amino acid glycine, there is a problem in that, and hence glycine becomes accumulated in your body. Okay, so that will lead to different seizures and alertness problem, because always there is more of inhibition because of accumulation of glycine. What happens? There is more of inhibition, so your body becomes slowed down. Your alertness is very less. Okay, you are not very alert because of the different surroundings which are there. So different sodium benzoate and other types of your drugs are used to reduce the effect of glycine, or to reduce the lower the amount of glycine, and hence improve the alertness. Last one is your glycinuria, where it is again a autosomal disorder, where it can cause a different reabsorption problem in the kidney. Okay, so that is all about glycine. So that's how your different diseases and different how your neurotransmitter or your receptor will work for glycine. Okay, am I clear with all this about glycine?